Hi, we're Dave Birchler,
And Caroline Brooks.
and we'll be discussing steroid hormone pollution and a novel method for determination in fish. Steroid hormones are cholesterol derivatives which act as hormones regulating gene expression in target cells. Androgens and estrogens control development of secondary sex characteristics, while progestogens prepare the uterine lining for ovum implantation. Glucocorticoids function in metabolism, but also inhibit the inflammatory response and enable animals to respond to stress. Steroid hormones are vital to bodily function, but why are we concerned about finding them in fish? Outfalls from wastewater treatment plants are often important zones of biodiversity due to the constant supply of organic material. Wastewater treatment plants are not designed to remove pollutants from sex hormones and corticosteroids, and nature's regenerative ability can no longer keep pace with the scale of organic compounds humans are dumping into the oceans. Surprisingly, some of the first studies on wastewater treatment plant impact on fish are from the mid 1990s. Since then, we've learned steroid hormones can cause altered sex determination, delayed sexual maturity, and decreased secondary sexual characteristics in aquatic biota in concentrations as low as nanograms per liter. The UN and EU have taken notice, and estrogens are on many environmental watch lists. Endocrine disrupting chemicals, or EDCs, interfere with endocrine systems. Although androgen and progestogen effects on aquatic biota have not been well studied, estrogen has been found to be an EDC affecting fetal brain development, leading to both hermaphroditic and unisexual populations. The image shows increased testicular oocytes in the smallmouth bass population exposed to estrone and estrogen. The genesis of the work producing this image was a series of fish kills, with EDC pollution the prime suspect. Steroid hormone pollution will impact humans through decreased food supply as entire ecosystems are disrupted via biomagnification of pollutant levels and a loss of biodiversity. To recap, we need fish for food, and they play an important role in aquatic ecosystems. Humans are negatively impacting fish by spilling steroid hormones into the oceans. In order to understand the severity and scope of the pollution, we need ways of testing fish for these steroid hormones. So how do we find out if we're polluting the fish? The authors of a recent study proposed microwave-assisted extraction and solid-phase extraction coupled to ultra-high performance liquid chromatography tandem mass spectrometry. As MAE is relatively new to this field, the parameters affecting extraction and analytical success were unknown. These parameters refer to microwave power, extraction time, and extractant solvent, to name a few. Prior research had extracted all families of steroid hormones in sewage sludge and a subset from fish tissue using MAE, SPE, UHPLC, MSMS. One of the major complexities here is the consolidation of the two preceding works, extracting these 15 different steroid hormones from the biological matrix of fish tissue. If successful, future studies could either refine the process or utilize it for studying steroid hormone pollution in new contexts. Thus, the aim of this work is to develop an analytical method for the extraction and evaluation of a group of 15 sex hormones and glucocorticoids of marine organisms. Once optimized, the authors tested the procedure on 2 species of fish. To understand why MAE, SPE, UHPLC, MSMS was selected, it is important to understand the complexities involved. First, the quantity of different analytes, 15 from 4 families of steroid hormones, presents a challenge. MAE was selected as an extraction method as it can quickly extract all analytes into the extractant, and liquid chromatography allowed those analytes to be easily separated. Additionally, MAE has high yields with less side products, compensating for the potentially low analyte concentrations. Finally, extraction from a biological matrix often yields proteins and phospholipids, which coalute, causing ion suppression and inaccurate mass spec readings. SPE removes much of these protein and phospholipid matrix components. So what exactly is MAE, SPE, UHPLC, MSMS, and how does it work? Muscle, viscera, gills, and skin samples from the Ghanaian puffer and the adorably named boop boops underwent homogenization and lyophilization to remove all traces of water, which is important due to how a microwave apparatus heats a sample. Next, through MAE, The analytes were dissolved into the extractant and purified through SPE before determination. A fairly standardized determination process is UHPLC MSMS, where analytes are separated through liquid chromatography and analyzed via tandem mass spec. Finally, the matrix effect was resolved using three internal standards, which is basically a way to estimate and nullify the impacts of non-analyte compounds that coelute. Laboratory microwave systems, like kitchen microwaves, produce heat through dielectric heating from dipolar polarization and ionic conduction arising from the microwave irradiation. The figure shows electric field oscillations rotate polar molecules. As the molecules attempt to align but fall behind the pace of field oscillation, resistive heating is produced. The efficiency of the EM radiation converted into heat is the dielectric loss. Similarly, the electric field pushes and pulls ions. As they collide with other matter, heat is produced. The ability of a substance to absorb electromagnetic energy into heat is the loss tangent, or loss factor, mathematically calculated as the ratio of the dielectric loss over the dielectric constant, which is a measure of polarizability. Each substance has its own loss tangent, and the loss tangent is frequency and temperature dependent. Scientists choose a solvent with loss tangent and solubility properties appropriate for their analytes. The samples in contact with the solvent are irradiated with microwaves, dissolving analytes into extractant. The theory behind the processes of UHPLC and MSMS should be familiar and are outlined in this figure. However, everyone knows practice and theory are two very different things. In practice, analysis can be skewed dramatically by what is called the matrix effect, which is basically all the other stuff you unintentionally dissolved in your extractant with your analytes. The matrix effect often presents with signal suppression during the tandem mass spectrometry, and in a biological matrix such as fish tissue, phospholipids are often a major coeluting signal suppressing matrix material. The exact mechanisms of signal suppression can be seen in the figure, but ultimately matrix components compete with the analyte for charge. Therefore, less analyte makes it through the mass spectrometer, causing signal suppression. To counter this, before running the extractant through the UHPLC MSMS, the extractant is purified in an SPE cartridge that separates the proteins and phospholipids from the analytes and extractant. There are many different SPE cartridges on the market, and they all work in slightly different ways.
As most of this study is focused on the optimization of the proposed extraction technique, we therefore must analyze each of the individual tests that were run to find the ideal conditions for which to complete the extraction. In total, 4 tests were run to determine these conditions. A 2-part factorial design was used to optimize the microwave extraction. A 2 raised to 4 factorial design studies 4 variables at 2 degrees. Here, 2 sample weights, 2 power levels, 2 extraction times, and 2 extractant volumes were studied. These components were studied to develop a general understanding of which kinds of factors consistently have the largest impact on extraction. Similarly, a 3 raised to 2 factorial design studies 2 variables at 3 degrees. Here, 3 power levels and 3 extraction times were studied. This test was designed to further refine the value set to have the largest effect on extraction. In other words, to find the actual quantitative values for these properties in which extraction was the most successful. A 3rd test was run trying varying solvents and their abilities to facilitate extraction while causing the least amount of signal interference during analyzation. And finally, a 4th test was run trying varying washes for their ability to further reduce the interferences that were mentioned previously. Now to discuss the results from the first round of testing. As touched upon before, this test was performed to develop a general understanding of which factors impact extraction the most. Here, we analyze the general impact these variables had on the extractant produced. In other words, these variables were tested for their ability to overall modify the extractants, not the degree to which they modified them. All runs performed in this test were random. That is, random combinations of the 2 possible sample weights, powers, extraction times, and extractant volumes were used in each round of testing. Power and extraction time were found to have the most significant effect on the extraction processes individually, but even more so when combined. In contrast, extractant volume was shown to have a negligible effect on the success of this extraction, and a value of 4 milliliters was settled upon as the ideal extractant volume. Similarly, sample weight was shown to have a minimal effect. Larger samples, however, give opportunity for more interferences to arise, giving way to lower overall recovery. Because of this, a conservative 100 milligrams of sample tissue was deemed optimal for this type of extraction. Here we have the results from the second round of testing, a 3^2 factorial design. This testing was conducted to refine the values deemed to have the largest effect on extraction and settle upon the values of which to set these variables to. Again, here all runs were random, where 11 trials tested random combinations of the various powers and extraction times. Additionally, it's important to note that powers over 700 watts and extraction times over 10 minutes were not tested, as these conditions led to solvent evaporation, rendering the extraction unreproducible. It was concluded that different target compounds seem to have different individual variables for which extraction is most successful. However, overall extractants were the most abundant and consistent when the microwave extraction was completed over 6 to 8 minutes at powers between 350 and 500 watts. Now to settle upon an extraction solvent. An effective solvent should be able to facilitate the extraction completely from start to finish with as little interference as possible. That is, the solvent refrains from revealing itself or other compounds in the sample during evaluation, ensuring only the compounds of interest are contributing to the data. Here, methanol, acetonitrile, and diluted 50-50 mixtures of the two compounds with water were all tested as solvents. First, we must see why we can eliminate acetonitrile as a possible solvent. Pure acetonitrile acted as the poorest solvent, yielding recoveries consistently under 15%, easily eliminating it as an option. The diluted acetonitrile may first appear like it could be a good potential solvent, given that it had slightly higher recovery rates than pure acetonitrile. However, the extractants yielded by this solvent held significant signal suppressions that were consistently over 30%. Therefore, both of these options were proven to be poor choices. The overall difference in extractant recovery between methanol and diluted methanol were minimal. Both options consistently yielded recovery rates over 50% for all tissues tested. However, diluted methanol was proven to be slightly better at extracting corticosteroids, megastryl acetate, and nandrolones, while pure methanol was better suited for estrogens and the remaining androgens and progesterones. Ultimately, the choice between these solvents laid in the hands of their signal suppressions. The diluted methanol often yielded signal suppressions twice as high as those yielded when pure methanol was used. Because of this, methanol was chosen as the ideal solvent for this method. With the previous steps ensuring success of extraction, the only thing left to do is analyze the extractants. However, it is important to ensure your sample can be accurately analyzed by your machinery. Here, the use of liquid chromatography mass spectrometry for the determination step was proven to be problematic, as the analytical signal was frequently at a loss due to interference from compounds present in the extractant. Especially problematic were things like phospholipids and proteins. Liquid chromatography mass spectrometry often will read for these compounds if they are present in the sample, which skews the results. Unfortunately, when methanol is used as a solvent, signal losses due to the presence of phospholipids tends to be higher. It is exactly because of this mishap that a wash step is necessary. The wash step chosen for this experiment was determined through analyzing signal suppressions after compounds were passed through various commercial solid phase extraction cartridges which are made to remove phospholipids and proteins from biological extracts. Compounds extracted with pure methanol and diluted methanol were both tested and washed with two types of solid phase extraction cartridges. Phenomix-free cartridges were shown to produce lower signal suppressions overall than the Oasis Prime cartridges, with best results yielded from those compounds washed with pure methanol, confirming methanol as the better solvent. With all variables settled upon, it's time to actually use this technique to analyze fish tissues. Guinean puffers and boop-boops found close to the wastewater treatment plants in the city of Las Palmas de Gran Canaria were tested for this part of the experiment. Across the board, muscle tissues were shown to have the greatest diversity of hormone compounds. However, these compounds tended to exist at very low concentrations across the tissues. These compounds were actually found at concentrations under the detection limits. Visceral and skin tissues were shown to have less compounds present, but at much higher concentrations as compared to muscle tissues. Progesterone was the only compound found present on all samples collected. They were found in concentrations up to 3.9 micrograms. Levonorgestrel and nalangolones were found in concentrations between 0.07 and 2.71 micrograms. Prednisolone was the only corticosteroid found in the samples. However, it was only found in boop-boops at an average concentration of 0.09 micrograms. And to much surprise, estrogens were not found in any of the samples collected. This is surprising considering this was one of the major concerns for the fish in the area. In conclusion, this study proves microwave-assisted extraction with solid phase extraction as the wash step and ultra-high performance liquid chromatography tandem mass spectrometry for the determination step to be an innovative and dynamic technique. This method allows for hormones and glucocorticoids to be extracted and identified in under 30 minutes with great accuracy. Additionally, it was shown to have great repeatability, yielding consistently accurate results during retesting. In regards to the relative recoveries obtained from this experiment, both species of fish analyzed were shown to have similar and at the very least comparable levels of hormones and glucocorticoids in their tissues. Despite the differences between these two species, the simple fact that they both inhabit and feed from the same compromised area seemed to be enough to drive them to absorb similar concentrations of the compounds of interest. Muscle tissues were shown to absorb the least amount of detected compounds, where viscera and skin tissues were shown to absorb the most. This is consistent with the results yielded from another experiment. In June of 2016, a team led by Ohelina Rose developed a slightly different method for the extraction of endocrine-disrupting compounds in fish, including many of the compounds that we focused on. Despite sampling different species of fish from the Bay of Biscay, an area near but entirely different than the area our study sampled from, this study also found that hormones seem to build up in muscles in lower concentrations than in visceral tissues. With a novel and effective new method for the extraction and analyzation of hormones and glucocorticoids in fish tissues, perhaps scientists will now begin to develop a greater understanding of how runoffs from wastewater treatment plants affect fish residing in the exposed areas, and perhaps even further down the line, how the consumption of these fish will ultimately impact the human body.